It's
Because
always
normally you are very busy, before.
no, that who is not, tell me.
Yeah, correct.
Everybody is busy, so you
Yeah.
have to make time. And I think in this pretext, you also sit and read. If it is an advanced lecture, then some reading would have been done, but this is
Yes, yeah,
basic. So,
yeah.
but so many things, tomorrow there is another program as a participating in some debate or something. So to prepare for this, to prepare for that, and then you become touch so many.
Okay.
Mm. So that's how it is. But I'm hoping it will benefit our students.
We
Only thing is
forgot
that
to, Chandra, I forgot to tell you that, there was in the ranking by which university? Stanford University. I
mhm.
am listed as in the top 2% scientists.
My God, lovely. Then you have to give me a treat, I say.
Yeah, sure. When I meet you.
I
And
know.
after I retired some 9 years back, but they have
Mm.
still taken my name also. Maybe towards the end, whatever.
My God, but that also is a great credit.
Yeah,
The top 2% is better than the top 3%. It is better than the top 10%. So,
yeah.
I mean, you always nowadays we look at what you call relative happiness. So we
Yeah,
feel happy. Our name being, pronounced or
yes,
visible
yeah.
somewhere, it's a lot
Somewhere.
of happiness, right?
I think they have taken data up to May 6th, 2020. Some citations and all those things. And they published an article in PLOS One journal.
I
PLOS
say,
One.
oh, plus one, may okay.
Yeah, but I have not seen. I didn't know also how they were assessing. But today I saw it, like somebody had written how they have assessed.
Oh, yeah, there are so many ways how people do citations and people who know this IT business,
Yeah.
they're really so smart, I tell you. Sometimes I feel, but you can't be everybody, right?
Yeah,
So,
right.
so I always felt, I wish I had known. Like I'm trying to join two videos and try to delete the between and add some, back effect. Like for example, even in Zoom, you can give a background which is different, right?
Okay.
Not this the dressing table and the
Okay.
balcony and all.
Yeah. Hawaii. I think my husband also knows. No, we can be in Hawaii or we can be in
Yeah,
some
yeah.
Taj Mahal or somewhere. No?
Correct, like that. So that is what I thought, but I didn't know how to do that. So
How do you do it, Bob?
Afterwards.
Afterwards, my husband will tell you how to do it. So
oh
next time you can be in
no,
Hawaii.
I tried it that day and I saw the background, but the background was so prominent that my face was not seen.
Okay.
So some green, this thing, coconut trees and all. But
Yeah.
after that,
It
then
distorts.
Yeah,
it
it distorts.
distorts. But I have seen many people not distorting also. I've seen many
Okay,
people having
maybe
the
that's.
nice background at the back and they are coolly sitting and talking. So I should try to do that.
Maybe
And
you can do
how to attach the video and how to insert something and all that I am trying. But I'm referring to so many YouTube videos to learn
Okay.
that
Good.
not confident still. Now today's is going to be
It's
one
time.
experience where you don't. It's time up, right?
Yeah, shall we start?
Yeah, I just muted.
Okay. So over to you, Nitana. Let's begin our today's session.
Welcome to all dignitaries, guests, and delegates. With great joy and immense exultation, I feel privileged to extend my warm welcome to all presented here for the Union Christian College Centenary Webinar Lecture Series 2020 to 2021. And dear participants, please follow the instructions during the webinar. Be sure to mute your audio so that there will be no disturbance during the entire session. Just a little housekeeping before we get started. If you have any questions during the presentation, please type them into the chat box in your Zoom or YouTube control panel. We will bring them up during the presentation. We will also have the discussion about all those questions at the end. And the feedback form of the webinar will be available only at the very end of the session. Today, we are presenting the webinar Stem Cell Medicine, which is presented by Dr. Chandra Viswanathan. She's a former director, Plasma Fractionation Center, and former head of Regenerative Medicine, Reliance Life Sciences, Mumbai. And also we have Dr. David Saj Matthew Sir, Principal of Union Christian College, Dr. Susan Ipan ma'am, she's a retired scientist of BARC Mumbai. She's also an adjunct faculty of Union Christian College, Aluva. Mr. Shyam Mohan sir, head of the Department of Biosciences of UC College, and all the faculty members presented here and all other active participants. The Department of Biosciences, UC College welcomes you all to the webinar, Stem Cell Medicine, which is organized by the Department of Biosciences, UC College, in association with UC College Alumni Association of North America. Stem cell therapy is the finest medical breakthrough in the history. It has bagged the much acclaimed Nobel Prize in Medicine in the year 2012. The miracle discovery, stem cell therapy, has lowered and firmly positioned India in the field of medical sciences worldwide. Stem cell therapy has changed the medical perception of incurable neurological disorders and has broken all shackles. The researchers hope stem cells will one day be effective in the treatment of many medical conditions and diseases. Right now, the most commonly used stem cell-based therapy is bone marrow transplantation. So, without any further delay, we may step into the official inaugural session. It is a mark of our undying tradition to invoke the Almighty at the beginning of any important function. So, like the great philosopher once said, function of prayer is not to influence God, but rather to change the nature of one who prays. Let's silent pray. Now, I would like to invite Mr. Shyam Mohan sir, head of the Department of Biosciences, UC College, to formally welcome the gathering.
Good evening, everybody. Today, we have Dr. Chandra here, and she will be talking to us on stem cell medicine. So it is a part of my privilege to welcome Dr. Chandra ma'am, the principal of our college, Dr. David Saj Matthew sir, and other dignitaries who are present here, as well as the participants and students. So without much ado, I'll just go into the formal welcome. Firstly, I would like to welcome Dr. David Saj Matthew sir. Sir has been the inspiration that has led us to conduct many seminars. The Department of Biosciences is very privileged to have him here, and hope he too is a part of the seminar for the full time. I know he is busy, and we have had a discussion earlier on this particular topic as well. I hope he finds time in. It's good that he found time in between to be part of this seminar also. I formally welcome Dr. David Saj Matthew sir to this program and for the inauguration as well.
Thank you. Thank you.
Now I would like to welcome Dr. Chandra Viswanathan sir. Ma'am has been the former head of the Reliance Institute for stem cell regenerative medicine, and she will give us an idea about how stem cell therapy can be utilized, and it will be a good foundation and understanding for our undergraduate students as well, and also for the students of the MSc Biotechnology and Bioinformatics. Very glad to have you here, ma'am, and I welcome you to this program. I would like to welcome Dr. Susan Ipan ma'am, who has had accolades very recently. Like she was just mentioning, we are proud to have you here as our adjunct faculty, helping our students to familiarize with the aspects of research and communications in research, inviting good scientific speakers, etc. I welcome Dr. Susan Ipan ma'am to this function. I would also like to welcome Dr. Seren Sarah John and Mr. Verghese Thomas, who have been coordinating this particular program. And I wish them all the best. And they have maneuvered the program in such a way that it enhances the participation of a lot of students from our college as well as from outside. And thank you for your efforts. And I welcome Dr. Seren ma'am and Verghese sir to this program. I would like to also welcome all the participants of this seminar from our college, from Kerala, outside Kerala, as well as international participants who are a part of this program. They're continuously supporting us by being part of this, and I hope you have a nice time here. Today you'll have a different session, and today's session will be on stem cell medicine, and I hope you have a good time here, and welcome you all to this program. Finally, I would like to welcome the teaching and non-teaching staff of our department. The cumulative efforts of all of the teachers have been instrumental in organizing programs of various proportions. And we have had a lot of sessions in the last 2 months. And it was a good opportunity to interact with some of the best academia and scientific. And it would always enhance our scientific perspective and help us to deliver the best. So I welcome all the teaching and non-teaching staff of the department and the college. And finally, I would like to welcome all the students of our college and the volunteers and participants from our college as well as from other universities and abroad. Welcome one and all. Welcome all of you.
Thank you, sir. Thank you for your earnest welcome address. Now, I cordially invite Dr. David Search Matthew, sir. Sorry. I cordially invite Dr. David Search Matthew, sir, Principal of Union Christian College, for the inaugural address.
Okay, thank you. Respected head of the Bioscience Department, Mr. Shyam Mohan. Today's speaker, Dr. Chandra Viswanathan. Our adjunct professor, Dr. Susan Epen. The webinar coordinators, Shavin Sarah John teacher, as well as Varghese Thomas sir. Other participants of this webinar and dear students. Thank you for giving me an opportunity to speak a few words about in this seminar. Of course, everybody knows that the stem cell research and stem cell medicine and is done wonderful things in the field of health science, as well as in many other areas of biological science. I'm not going deeper into that one. We have a very eminent personality who knows very much about this. That is Dr. Chandra Viswanathan, and we are privileged to have her here in this webinar. So I'm using this opportunity to welcome Dr. Chandra to Union Christian College as principal. And I'm sure that this seminar is going to be very useful to the participants because of this. The topic is very relevant in this field of medicine as well as in other biological sciences. So I'm not standing in between Dr. Chandra and you. With these words, I wish to give all the best to this seminar, as well as congratulating the organizers for conducting this seminar. Thank you. Thank you very much.
Thank you, sir. Now, I would like to invite Dr. Susan Epen, ma'am, retired scientist of BARC, Mumbai. She's an adjunct faculty of Union Christian College, to kindly introduce our chief guest into the webinar.
Good afternoon. I am extremely delighted to introduce Dr. Chandra Viswanathan, former head Regenerative Medicine, Reliance Life Sciences, Mumbai. She was also former director of National Plasma Fractionation, Mumbai, and also medical director of Cord Blood Banking Initiative, 2002. She has done her MD in pathology from Mumbai University and later did her PhD in applied biology also from Mumbai University. She has 2 diplomas to her credit, diploma in pathology and bacteriology and diploma in transfusion medicine. She has done a lot of research and she has achieved quite a lot. Some of the achievements are as follows. She has developed affordable blood products for patient population. She has set up the first blood component way back in 1986. She has set up blood banks, component laboratories, and she is a specialized person in laying down quality systems and developing and marketing blood products. She has a large number of publications to her credit. She has 80 publications. Dr. Chandra has mentored 15 students in pathology and transfusion medicine for their MD degree. She's an excellent speaker at several international forums. And the focus of her research is on stem cells as a regular prescription medicine. Dr. Chandra has the grit and determination to create more milestones. She sums up saying like this: success has become my habit, and I have been very successful in any new venture that I have undertaken. She is also an exponent in Carnatic music. She wants stem cells as a regular prescription medicine. So today, she will share with us her ideas on stem cell medicine. Dr. Viswanathan, please.
Am I audible?
Yes, ma'am.
Yeah. Very, very good evening to all of you. And I must, at the outset, express my deep gratitude and happiness in being a part of this very, very interesting program. Dr. Susan Epen has been my dear friend, and she has, I did not realize that she was on the top 2% of the scientists in the world. The results or the information of which I just received a couple of minutes ago. So we are talking about Mrs. Susan Epen, who is herself a very big scientist. My thanks are also due to the Aluva College, the organizing committee of this the celebrations that you are going through, because I heard that the college is celebrating the centenary year, and you are inviting people from all walks of life to address your school, your students, your undergraduates, and also the non-academic members. So it's such a great initiative, especially in this lockdown period when you're getting ready to accept, ready to consume information. I think there cannot be anything better. And I sincerely hope and feel that this effort does not go waste, because in the next 45 minutes to an hour, I will try and talk to the undergraduates of the college, try and give them a flavor of what stem cell is about, or what stem cells mean, and what they can expect, as you move forward. So people have been hearing about stem cells for quite some time now, and a time has come when they want to actually see whether this can actually be translated onto the bedside. So that is the kind of expectations our doctors, our patients, the families are all expecting. Having said that, I also would like to express my gratitude to all my former colleagues, former junior colleagues and friends who have helped me come to this level, because unless everybody works together, these kind of results, this kind of information, this kind of knowledge doesn't accumulate. So I really remember my former colleagues, and through all the ups and downs that we have gone through, to be able to share the experience that I'm going to share with you today. So once again, I hope I make a difference at the end of 1 hour. So can I start with my first slide, please?
Ma'am.
Yeah, okay. Let me just go to the first slide. Yes, the first slide is here. I just want to. There are too many things coming in the screen, so I just want to. Yeah, is it clear now, please?
Yes.
Yeah. So I, as Dr. Susan mentioned, I have been very lucky to have been a part of developing cost-effective solutions to the patients. And this has been in the area of plasma-derived blood products. And in the recent 15 to 20 years, it has been in the area of stem cell medicine. So we planned, we conceived, we conducted several clinical trials in the hope that these would graduate and come to the patients on a regular basis, so that it becomes a prescription product. And as a part of this journey, I had to interact with large number of regulators, young and old, people who did not know what stem cell was. So it was a sort of a new learning to be able to lay down certain rules that did not exist, to form a framework by which all people in at least within the country will follow, so that we are all on one platform. Because as I speak today, there have been developments year on year since the year 2000. As a part of this, therefore, there was a lot of exposure that I gained with regards what is the meaning of quality, what is the meaning of protocols, what are processes, and of course, definitely there was a palpable difference that we could make as a part of the organization, where we could we could actually do cord blood transplantation by offering the products that we had for some children with thalassemia and allied congenital disorders. So these were some of the experiences that I have had. Now going to the talking to a group of undergraduates who are extremely excited. They want to know what I'm going to speak, whether it's going to make a difference to them, or whether it's going to make a difference to them, either their families who are having some problem in terms of perhaps somebody in the family, unfortunately suffering from a disease which is otherwise not treatable through the traditional medicine that is available today, or because the undergraduates are looking forward to some career options, or to see whether all this stem cell medicine is only just a fiction, or what else is in store for us. So these kind of lot of expectations are there in the minds of people who are listening at this point of time. So if you just focus on this slide, I have just written a couple of points. When I talk about this, say for example, designer babies, or I'm talking about a heart beating in a Petri plate, or making a baby in a test tube, or can we produce a baby which will never have a cancer, a certain type of cancer, or a baby which will never suffer from an HIV? Or is there some kind of a magic bullet by which I can stop aging, which is a big curse? Is it possible that the same sex couple can have their own baby, their own biological baby? Or can I have a baby with a certain selective brain capabilities and a certain cosmetic capabilities, or say beauty with brains, etc.? So all these we have seen in movies, and we thought 20 years back, all these were just a matter of fiction, and therefore we passed it off. But now in the last 10 years, 15 years, since the year 2000, there have been so many research papers, so much of evidence-based medicine which is staring at our face to say that all these were possible to a small or to a very large extent, till it becomes to an extent that is truly repeatable, that is truly dependable, that is truly minus all the problems or the side effects. We cannot still say that we have already reached the goalpost. So we have yet to reach the goalpost in many aspects, whereas we have almost reaching the goals in a certain few other areas. So what in this slide is to show that it is no longer a fiction. It is doable. Some of them are almost on the way to reach, and some of them have been already successful. For example, a baby which is produced in a test tube, what we call in vitro fertilization, was once believed. If you remember, people will know that these used to be discussed in Ramayana or in our mythological stories, where, suddenly from nowhere a baby was produced, or where was the sperm, where did the egg come from, but you had 100 babies being produced and somebody was produced from thin air, etc. So all these were stories, but no longer they are stories. They have been done, they have been shown, they have been documented, and in vitro fertilization has become a part of regular practice today. Having said that, I'll move on to the next slide, where I'm just going to try and see. I'll introduce the subject, try and differentiate, try and convince, or try to elaborate on what are the basics about stem cells, what are the current perspectives in research, what are the unmet medical needs, and what is it that really bothers us today in terms of clinical requirement. And people may ask you questions about what is the status of research today, therefore where have we reached. So we'll try and address some of those questions. what are the perils, what are the potentials, and therefore what are the challenges we face for us to be able to move to the next slide, to go to the next milestone. I am not going to be able to cover these areas in an organized manner because there is a hidden amount of potentials in practically all the slides. So I would be trying and covering, and I'll try and summarize as often as possible so that the undergraduates or the people who are not exposed to this in the past, my sincere apologies to those people who are experts, who are already in the know-how of things, especially the stem cell area. I'm sure there are many amongst you who already know about this. So it could be a repeat. So my apologies to those people. So coming to the story of stem cells. So we all started with one cell. All of you know this. So either we started with one cell. What is that cell? Maybe we started with the formation of a gamete where the sperm and the ovum joined together to form what is called the zygote. That formed the foundation of you and me, and that is how we started. And from a few cells, the cells started enlarging in numbers. It just went from 2 to 4 to 8 to 16 and so on, right? And when it became a full-blown body, and that's what you and me are today. So from a very small number of cells, we have been able to form a very complex organs and a human being like all of us. The right side of the picture is saying that the main stem or the main source is the zygote, and from where we as a tree are standing to show that we have been able to evolve and form all organs of very highly complex nature, the heart cells, the brain, the spinal cord, and then, so God is the best architect of you and me. We have not been able to emulate him, but all we are trying to do is to at least understand what he has done first, then alone playing with it, tweaking it, or trying to juggle with it and try and see whether we can offer something in the form of a treatment for our patients. So stem cells, therefore, the original cells, the ones that are going to replicate, we are going to define it once again in a little better way so that you understand better. So stem cells are these are cells which are able to repair or replace damaged tissue. So in this branch of medicine is called regenerative medicine, which is just about 2 decades old, or maybe 2.5 decades old. Earlier, it was not called as regenerative medicine. This term got coined a little later on, maybe say 1995 and later. And in this regenerative medicine, what is it that we are going to do? We are going to be able to give some healthy cells so that we can repair something which is damaged, or trying to reverse certain diseases, or trying to undo some injuries, or trying to see whether we can heal certain injuries. Now, these injuries can be small, they can be very large. They can be a little small, like say something like a wound, a non-healing ulcer, to something like diabetes, which is an endocrine problem, or to coming to a series of cardiovascular disorders, or to treat something like a cancer, which is now the big C word, as we call it, is we don't want to touch that subject at all because the subject is so huge. So we are going to say that what we want to say is regenerative medicine, therefore, is that branch where we are going to be able to give normal cells and many other things along with it, so that we can actually replace damaged tissue. We can reverse certain diseases. We can reverse or repair or heal injuries. So this is what I just want to leave you as far as this slide is concerned. Stem cells have some unique capabilities, which is what we are going to say. What are we going to see here? We are going to use also certain terminologies which are less often used, but for the sake of understanding, I have tried to retain these terminologies in my slides. Now, coming to unlimited stem cells. Now, unlimited stem cells are obtained from certain sources. Now, which are these unlimited sources? The best source is an embryo. Why is it an unlimited source? Because from an embryo, which was otherwise going to be potentially forming a full-blown baby, it has so much of potential. The inner cell mass, which we are going to show in the next slide, we will try to elaborate on it as we move forward. So the unlimited stem cells we are now going to derive from 3-day-old embryos, which are otherwise thrown out. I'm not talking about those embryos which are going to be used for the sake of formation of a baby as a part of in vitro fertilization. I'm talking about those embryos which are not going for the in vitro fertilization experiments or for treatment in the in vitro clinics, in vitro fertilization clinics. The word limited stem cells are found in practically all organs or all tissues. They have been successfully used in bone marrows and skin and hair transplants. Why? Because we're going to study why these limited stem cells have been useful. We'll go forward, and so these two terminologies I just want to introduce here. There's one more terminology called fetal stem cells, where the stem cells are derived from the fetus. So suppose you have a 10-week-old or a 15-week-old or a 20-week-old fetus, because it is already stillborn or the mother is not able to carry on with the progress with the pregnancy. If at all she aborts and then you get the fetus out, these fetal stem cells have got very good potentials. It is not as potent or it is not as versatile as the embryos, but certainly it forms the focus of it. It gives you a very good raw material for you to study. So these fetal stem cells have a lot of capabilities. Then comes the next order of cells, which are called as cord blood stem cells, where the baby is fully born and the leftover cell from the cord blood is another type of cell. The cord blood has got certain cells, which a small portion of it are stem cells, which have certain unique function. So here we have learned 4 terminologies like unlimited stem cells, limited stem cells, there are fetal stem cells, and there are also cord blood stem cells. Now, fetal is coming from the fetus, cord blood is coming from the cord tissue from a baby which is already born normally and is already handed over to the parents. So cord blood stem cells are also unique because they have certain potentials. So coming to the next, as we move forward, I'm just wanting to recall what I said a little while earlier. Now, as you can see in this slide, here we have the blastocyst, and you have a group of cells here. Now, this is all what is going to form the future baby. And these are the cells which are going to form the extra embryonic tissue, right? Now, from this tissue, suppose I had my hand on this, if I can lay my hand on this, and I know that I get it in the laboratory through some means, which we will discuss a little later. If these inner cell mass cells can be taken out, they have great potentials because this is what was supposed to form the future baby. And therefore, you can theoretically, and has been proven in the recent past, in the last 2 decades, several researchers, several scientists have actually proven that from these cells, you will be able to successfully derive neuronal cells. You can form some other cells, you can form some other organs, practically everything, but not the entire baby, because we are only using it as a part. So these are part of the cells that we have used from the zygote, called from the inner cell mass of the blastocyst. Going a stage earlier, suppose you had laid your hands on the totipotent cells, which is before this stage. This is called the totally potent, right? Fully potent. So it has the full potency. That's why the word totally potent is used. So going backwards, therefore, an oocyte is formed, and from here totally potent zygote is formed. From the zygote, we go to the blastocyst stage, which is the future human baby. From this, if you had your luck and you had your chance to study a research, or if you could do research on inner cell mass, you are going to be able to study these things, right? So now we are going to introduce a few terminologies like totipotent, which I already mentioned to you. Now coming to pluripotent. If it is not this, if it is only partially potent, that plural is a few more tissues can be formed. So they are descendants of totipotent cells, and they can differentiate into cells which are derived from 3 different germ cell layers, namely the mesoderm, the ectoderm, and the endoderm, right? So all the 3 germ layers, you will be able to experiment, you will be able to research successfully if this was your starting material. Now coming to multipotent stem cells. Multipotent is one step lower. It's not as proficient or as vibrant as the earlier step, but this multipotency it still has potency, but it will form cells related to its own family. We'll come with those examples. Now the examples of this, to cite here, we'll talk about hematopoietic stem cells. So something to do with, it will form cells, all the cells that are related to the blood forming system. Or there could be all neuronal cells will be formed from a certain area, or something coming from the endoderm. So there will be a limitation in the potency, but still retains some potency. Therefore, coming down, still lower down, you go to unipotent cells. I hope all of you are with me on this. And if there are any questions or lack of clarity, do wait for the talk to be over, so that I'll be happy to address any questions that are less understood. Going to the unit cells, normally means one. So as we move further downwards, the potency limitations, the limitations of potency can be very visible, and therefore you come to a stage where you will form just one type of cell. For example, I pick up a cell which is a stem cell from the heart, or I pick up a stem cell from the deep down tissue of a muscle, or I'm taking a stem cell coming, say, from a skeletal muscle, or coming from a pancreatic islet. So these mean that it has potency only to form that particular cell. So what has happened over time, as we differentiate, as we go from here downwards, the capabilities come down, the specialization becomes more and more sharper. So the typical analogy would be, say, for example, if you were born here, you could, suppose you put yourself in your career path, you could have become, a doctor, a scientist, a lawyer, you have all capabilities, but you already taken the science stream. So you do not, I don't think, at least now with the new education policy, things may change, but I'm using this analogy because you and me can resonate on this analogy very well. So if you have already come to the science stream, so you know that you have limitations. So you are going to take science subject. You are either an engineer or a doctor or somebody to do with sciences, right? From there, if you have already come into BSc Biosciences, then you're going to be able to work only in this bioscience area. So these limitations are, as you move forward, you start focusing on what you can do better and better and better, right? So now coming to certain characteristics. Now these characteristics, one is the characteristic is self-renewal. So the ability to go through numerous cycles of self-division. So the cell division keeps going on and going on, but it maintains the undifferentiated state. What you would expect is, suppose a cell is dividing, it will get exhausted, and a day would come that I don't have any more of the raw material. It doesn't happen so, right? So there is always a maintenance of the stock. The stock is always getting maintained. The stem cell stock is remaining there. And the capacity to differentiate into any mature type is retained. So what happens? A stem cell will therefore divide and form one stem cell and form another daughter cell, which will do the function of differentiation or specialization, whereas the basic cell, that the other stem cell that it has produced, will continue to be the stock, right? So I'll elucidate this using a small schematic representation here. So we are using, this is symmetrical division here, as you can see here. Here a single cell is giving rise to two identical daughter cells, and then so the identical daughter cells are endowed with all the stem cell properties. So there are two stem cells that are formed. Whereas in an asymmetrical division, I repeat, in an asymmetrical division, you have one stem cell and then you have another progenitor cell. So this progenitor, depending on which organ it is in, it could be either in the bone marrow, or it is in a soft tissue, or it is in the brain, depending on where it is, it will start determining its fate, right? So this stem cell will continue to remain a stem cell because the line will continue. So there is no exhaustion. Otherwise, a day would have come that we are all exhausted. Some of our organs get exhausted. Typical example, a bone marrow. It is alive throughout our life. Till the time you die, the bone marrow functions, because what is the business of the bone marrow? The business of the bone marrow is to form blood cells throughout your life. The day the blood cell stops functioning, the bone marrow is shut down, either because it is slowing down because of a disease, or because of a cancer, or because of a drug, or because it is occupied by some other tumor or whatever. Till that day, the bone marrow is going to form blood cells. So the moment your blood cell production is affected, because the bone marrow is affected directly or indirectly through disease, that is when you need to worry. That is when doctors intervene, right? Going back to this explanation, these progenitors will go through multiple progenitor division and it will mature to the cell that it is expected to form. So if it is going to be forming a cardiac cell, it will form a cardiac cell and it will become a mature cardiac cell. As mature cardiac cell degenerates or regenerates or whatever, we are going to know what is going to happen if these cell stocks are gone, is what is a question that comes in the minds of all of us. I'll try and address this question too. So if I have made myself clear on this schematic representation of what I've tried to show, to explain to you what is a symmetric, what is an asymmetric, and what are progenitors, I think I will move forward to the next one. Now coming to the embryonic stem cells. So what are embryonic stem cells? A word which all of you have heard. For those of you who have not heard this, these embryonic stem cells are the same inner cell mass, the cell line that comes out of the inner cell mass, which is very immortal. It doesn't die. Why does it not die? Because we want to keep it that way. We want to keep that living all the time. What does it mean? It means that these cell lines, this embryonic stem cells, a group of cells, will remain in the laboratory in that stem cell situation because we don't want it to progress and we don't want it to become an adult cell. Why? Because we want to study. It has great capabilities. It can form 200 plus type of adult cells. So that forms your stock. So you want to keep it in the stem situations. The stemness, as we call, the stemness should not be disturbed. So we try to keep it in that stage by keeping it in certain situation. We keep it in ultra cold temperatures, and we do not allow any environmental changes or any milieu to change for it to actually progress and become a mature cell, unless the researcher wants to do that, right? So this forms a very big resource for regenerative medicine because, as I alluded to earlier, we have these cells that can form all types of cells. Now now coming to the ethics of ESC, we'll touch a little bit on this. Now, if embryonic stem cells have been studied, I also want to tell you that embryonic stem cells were studied even the year right from 1960s onwards, but it's only in 1997 that the first stem cell line was actually established by Dr. Thompson in the in his Wisconsin laboratory. Until that time, there were a lot of research papers, but this first stem cell line became a very big milestone in regenerative medicine. And when this stem cell line, or what we call embryonic stem cell line, came up, a lot of discussion started, a lot of discussion, or as to how is it that you can take this forward, because it has so much of potential. So if I were to make neurons in the laboratory, if I were to make heart in the laboratory, it's all doable, right? But at what cost? So came the biggest questions about how is it you can procure blastocysts from the in vitro fertilization clinics, or those gametes or those zygotes which otherwise are not useful for the family. Having said that, there's a lot of ethicists, a lot of political discussions, a lot of religious discussions, and a lot of other discussions came up as to where there could be a lot of exploitation around this, which is evidently possible, right? So in a country like that of ours, suppose I tell someone that somebody can sell their unused zygotes or unused blastocysts, which are otherwise could have actually formed a baby, and you can sell it or somebody is going to misuse it. We don't even know how it's going to be used, right? So therefore, the ethics around, is it justifiable to use blastocyst for these purposes? So it's a very big discussion, but I just want to leave this slide and also the thoughts in your mind to say that it's not exactly very easy for us to discuss. There is also one more slide where I'll discuss more about this ESCs. If that was the case, that in 1998 you had one stem cell line, and afterwards more than a dozen stem cell lines came up, people started investing on this research. That is how regenerative medicine started getting popularity. They thought that it will become the next milestone, it will become the next new medicine, it will become the next new revolution in medical sciences. But having said that, the fear about exploitation, the fear of the ethics around embryonic stem cells prevented large number of investments, including that of governments. The governments did not allow too much of investments, including that in the United States now or in other countries as well. I'll move to the next slide. I hope I have been able to talk a little bit. I have been able to convey something about the embryonic stem cells here. So I'm going back to bone marrow transplant to tell you that stem cell is not a new issue. It's something that many people knew about. And in 1956, after having known of the fact that the bone marrow is a very large organ, it's a very complex organ, and any bone marrow disease can give rise to problems with hematopoietic, or it will land the patient with hematopoietic problems. And therefore, to be able to offer it as a treatment, the first bone marrow transplant was formally conducted in 1956. And after that, after a gap of so many years, embryonic stem cells were isolated from mouse blastocysts. That forms one of the other milestones, because researchers always use smaller sources like the mice or zebrafish and whatnot. So ESCs from mouse blastocysts was a milestone. In 1988, the characterization was a very important point I want to highlight. They came to know that if at all you have the embryonic stem cells or hematopoietic stem cells, say in 1988, hematopoietic stem cells got a lot of mileage, a lot of attention. Why? Because at that time, they came to know that bone marrow has a lot of stem cells, and some stem cells which are very particularly carrying the CD34 positivity, which has certain other characteristics, are those very important cells which form hematopoietic stem cells, which generate red blood cells or the white blood cells or the platelets. So these CD34 cells, the CD34 positive cells, were very important. So this was a very landmark understanding in 1988. And then came hematopoietic stem cells that not only were isolated from bone marrow, which we already knew about, the hematopoietic stem cells were also isolated from cord blood. I told you about cord blood just 2 slides earlier. Cord blood is the leftover blood after the baby is separated and handed over to the mother. This forms a very important source where you can have a few stem cells. It's not as if 100 on 100 cells are CD34 or they are positive for hematopoietic stem cell characteristics. Very small percentage of them, a very, very small percentage of these cells are the ones that have these characteristics, that have the potency to form blood forming cells, right? So it is Elaine Gluckman in Duke University who did the first cord blood transplant, because there were challenges with bone marrow transplant. Bone marrow was not as easy. So it was understood, or it was thought, that if at all we get an alternative source, therefore cord blood became the next important source. Bone marrow being very difficult to get, or we don't get a match. We are not touching about the problems related with bone marrow at this point of time, but cord blood became a simpler solution, easily available. So many mothers can actually donate. So therefore the plentiness, the availability, and the lack of ethics, nobody needs to steal it, so you can have a consent form signed. And the peculiar characteristics of these stem cells, because they could be very easily made available to somebody, because it's already available in the laboratory. You don't have to hunt for some source. So those positive points became very attractive, to the researchers. And therefore in 1988, when the first cord blood transplant became successful in a child with Fanconi's anemia, in fact, that child is alive even today, and he has got married and he has got a child. So that's what I know about the first cord blood transplant that was performed by Elaine Gluckman. So this 1988 experiment on using a cord blood transplant became a very, very important point. Therefore, yet again, they started saying that let's start and bank these stem cells from the cord blood so that it can be useful as a medicine in future. I leave you with that thought and I'll go forward. 1992, stem cells were isolated from the brain. That became yet another point where somebody thought that once again we have some stem cells to work with, whereby we would be able to differentiate and specialize it and perhaps form cells of our requirement. For example, suppose somebody has a motor neuron disease or a Parkinson's disease, is it possible that I can produce these cells? So that forms the thought, that was a thought process. I already told you about the ESCs, the first the embryonic stem cell line was formed by James Thompson in Wisconsin. Afterwards, of course, a dozen cell lines were formed. The research had its own share of adventure and problems because of the financial resources. And there was an embargo on who can work on ESCs and who cannot and things like that. So just to tell you this was only to tell you that from the bone marrow experiment, which was a success, which continues to be a success today, which is a line of therapy even today for large number of blood cancers and blood disorders, we embarked upon cord blood as another source of stem cells, which was also a landmark observation. Therefore, want to tell you that the advances in science was, if not in leaps and bounds, but it was very appreciable, was something which was noteworthy. Okay, so now coming to what are adult stem cells. So that means, as can be seen here, I just want to tell you that adult stem cells are the stem cells that are formed in adults, as the name signifies. It is in all tissues, practically found in all tissues. We will study a little detail. It is found in bone, cartilage, in the skin, in the nervous tissue, in the mucosa, everywhere, right? And specific multipotent cells have got these cells which are present in adult tissues, which are buried in adult tissues, have certain potencies. And why not exploit these cells if at all they do have some kind of goodness, some kind of characteristic? So the thought started going towards working on adult stem cells, because they knew that embryonic stem cells had certain limitations despite the fact it was versatile. And therefore, people started finding out how to study these cells and how to actually replicate all these experiments in mice or in rats, etc. Because everything that we do in humans will be done only provided it is proven in lesser animals or in mammals, etc. So that's how we always go. There's never an experiment in human beings directly. So coming to one more concept that we had earlier, we used to think that if at all somebody has a liver injury, somebody has a brain injury, it is once you have a stroke for life. Once you have a liver disease, you have a liver disease for life. You can do nothing about it. But the science has changed so much in the last couple of years. We are able to see that we are able to actually regenerate. We are able to form brain cells in the laboratory. We are able to form liver cells in the laboratory. We may be able to do some tweaking of this experiment so that it can be gainfully used for patients with end-stage liver disease, with end-stage brain disorders, etc., etc. So that's the line of thinking. So I wanted to tell you this one slide in brief. So because embryo has got certain moral status, and there are a lot of issues with whether we are going to be destroying embryos, etc., despite the fact it is good. So we are going to ignore embryonic stem cells for the time being, and we are going to move forward. And I understand, or I feel that maybe you have got this concept clear. So therefore, we are moving further with what are adult stem cells. So with adult stem cells, we do not have any moral ambiguity. We do not have any legal issues. For all you know, your own body can donate for yourself, right? It is possible that your own liver If at all I tell you that there are liver stem cells in your own liver, it is possible that at one time in research, we can have our own. So I can be my own donor, which I put up in the first slide. It is no longer a myth. It could be a reality. It is almost closing, or we are nearing reality. So these possibilities are abundantly available with adult stem cells. So where are these present? These are present in the peripheral blood, as I told you, because the blood is a liquid tissue. It runs to all the parts of the body. So you can derive stem cells. You can actually extract stem cells from the peripheral blood. You can extract stem cells from the bone marrow, which is the best and the most beautifully studied of all organs. I told you about cord blood. I'm also going to tell you that there are going to be a lot of tissue cells. There are a lot of stem cells present even in the cord tissue, in the peri-birth tissues, that is like placenta. Like you can have even amniotic fluid in the amniotic sac. In all the areas, there are small patches of stem cells which you can extract. But depends on whether you want to focus your attention on that, or on cord blood, or on cord tissue, or on bone marrow. So it's up to the researcher to decide which area is his area of interest. On the left side, you can see that the entire body is available for you to do research. You can pull out a tooth from the dental pulp. You can pull out dental stem cells. You can pull out some cardiac tissues. You can pull it out from liver. The difference between what you see on the left, some examples on the right, are these are all more deeply buried, whereas adipose tissue is more easily available. From endothelial progenitors, from blood, more easily available. Bone marrow, easily available. Skin, you have plenty of skin around you, so you can study it so well. So that's what I wanted to show here. One other thing. Okay, let's close this slide also. So here is what I want to tell you, in case you have some questions about what is the evidence. So we are talking about evidence-based medicine at all times. So when I say evidence-based medicine, in this case, I'm trying to show that in the hematopoietic stem cells, which are present in the human bone marrow or in the peripheral blood, we have documented evidence that bone marrow produces lymphohematopoietic regeneration throughout life. There are another group of cells in the bone marrow which are called as mesenchymal stem cells. These mesenchymal stem cells not only are present in bone marrow, they are present even in cord blood, cord tissue, etc., etc. And these can also produce these cells. This is what the recent experiments have shown. You will be able to form neural. We have already proven that we can produce neural cells. We have formed cartilage cells. We have shown through experiments that we could form bone tissues. The neural stem cells, therefore, have formed by, there is evidence, there is research, there are research papers, enough and more of them, to show that neurons, astrocytes, oligodendrocytes are all produced. So with all these getting produced, now, I don't think the list is so exhaustive, but for the sake of interest, I'm just wanting to say that stem cells of the intestinal epithelium. Now, intestinal epithelium, these are the stem cells which are present in the base of each crypt. Each crypt of the, the small, like villi, right? At the base of these, they are present. And these form the Paneth cells, brush border. So you should not be surprised that even if somebody has got a lot of intestinal problems, some loose motions, and you lose a lot of your intestinal epithelial cells, the stem cells are going to produce these cells for lifetime. So the other example of cells like bone marrow is the intestinal cells. For lifetime, you will form newer and newer cells with no problems whatsoever. So the other two very, very lively examples are the skin, where you keep brushing off your skin every day during your regular bathing. New cells from the bottom of the epidermis is forming new cells almost on a regular basis. So this is the beauty of nature, and this is God given. So these labile cells are going to be able to give you the stock of cells that you need for your day to day, right? Now coming to the mesenchymal stem cells, is a type of adult cells which you can bet on, because these cells have been very well studied by researchers world over. It is the bone marrow mesenchymal stem cells that have been very, very highly studied or maximally studied. And we as a group, in my earlier appointment, I have studied mesenchymal stem cell from cord tissue, from cord blood, etc. And we have been able to prove, as like many other researchers world over, that we could form large number of those differentiated tissues, be it the the insulin secreting cells, or the chondrocytes, or what you call cartilage cells, or the bone tissues, etc. Why? Because these cells, which are mesenchyme means these are the intermediate cells, or these are the cells which are present in the pulp of the bone marrow, buried into which are the hematopoietic stem cells. So this is the base, this is the carpet, this is the something like a bed, which actually gives rise to all the cytokines, or the chemical reactions, or the right environment that is provided to the bone marrow, so that these stem cells actually produce what they're expected to produce. So the mesenchyme, or the below, or the background cells, are the ones which have the maximum characteristic. They have equally good potentials, and these are the cells that perhaps is the interest, or it's forming the focus of interest of several researchers. And like us, mesenchymal stem cells from these tissues are being studied. Now, the beauty between the difference between mesenchymal stem cells from you and me, that is from the bone marrow, is different from mesenchymal that are derived from umbilical cord, because these are all very naive, these are all new, these are all coming from a neonatal tissue, these are all coming from a child which is just born. So the capabilities are a little superior. Okay, we won't go into the details, but for the understanding of this talk, just want you to remember that the mesenchymal stem cells derived from these sources are more naive, easy, but equally versatile. So we are going to for those of you who have this curiosity, the curiosity of as to what is it that, how is it, how do these cells look? Just a flavor. So single cells under the microscope, if you put it under the right microscope environment, you will actually see these cells under dark ground. You will actually see them glistening like this, single cells. And if you want to see some colonies, you will see the mesenchymal stem cell colonies here like this in the Petri plate. They are actually stuck, and you can actually see them in and this is how if at all it forms a carpet, they look like fibroblasts, they look like elongated cells, and these cells have to be characterized. And for you to be able to prove that these are the cells that you believe they are, you need to show that they bear certain characteristics. That is how you will try and prove your science, right? As I told you, so cells in the laboratory, we can convert these cells by giving them the right type of cues. We say when you say cue, the right type of signals. So we give them the type of chemical signals, or the right type of cytokine signals, as we call it in technical language. And these can be converted into adipocytes, or the fat cells, and glial fibrillary proteins, that is the neural cells, etc., etc. So the immediate question that comes to your mind is, it's so easy. If that is so easy that you can you can form some bone. So if somebody is having a bone deficiency, I can produce this and give it to somebody. Yes, theoretically, all that is correct. And if somebody has got, so nobody is in need of a fat cell, because I just wanted to prove that these cells are forming fat. Now, what were these experiments? These were to prove that mesenchymal stem cells can not only form tissues of the mesodermal lineage, they can also form of ectodermal lineage. So that versatility is what we have proven. And we have been able to show that if somebody wants some cartilage cells, somebody has got a defect of cartilage, it is doable, it is possible, right? Okay. Now coming to the cord blood banking. So the cord blood banking in the regenerative medicine landscape became very popular since 2000. The 2001 end or early 2002 is the first public cord blood repository which was set up in a private company that I work for. And this was the first landmark in this country, the public cord blood. When I say public, it means it can be accessed by anybody and everybody. So large number of children or their moms actually donated the newborn's cord blood because of the usefulness of the cord blood. And it used to be kept under liquid nitrogen in a machine like this under ultra low temperatures at minus 196 degrees temperature, etc. And these are all the cycle of events that we go through before we actually bank them. And these cells are immortal. See, once you have extracted the cells from the cord blood and kept them in a small packet so that it can be kept for future use. So it's just a very small size packet. How small is it? It just looks like this. So this is the way it is kept. It is cryopreserved in a steel canister. Steel canister gets into liquid nitrogen, and liquid nitrogen is bathing all the cells throughout. At no point of time in its life cycle, you will have a shortage of liquid nitrogen. It gets continuously monitored for its temperature, for its variations, and you keep on instilling liquid nitrogen from outside at all times. So it's a business where you have to be on the vigil, you have to be on the watch. There's a lot of investment, and you have to keep proving to yourself that the quality of these cells are good. The quality of the system is good. You have processes, you have procedures, you have technologists who have to do something day in and day out, right? So these kind of processes were established, and many other companies, a dozen other companies followed suit, saying that there is a lot of potential, and that's how cord blood became pretty popular, like it became popular in other countries. Now, having said that, we are now going to the next level of, so how do they work? If that was the case, now I am assuming that you have understood that stem cells are of 3 different types. I have yet to talk to you about another cell type, which I will come a little later. But before we come into that, what I have tried to convey so far is that embryonic stem cells are most versatile. If I had the choice, I would work with it. But I am so worried about the embryonic stem cells associated problems of ethics, morality, availability, and things like that, and the funding, therefore, associated with it. Therefore, you choose to go to a next line, which is called adult stem cells. And I also told you that adult stem cells are found in immense measure in you and me. So you necessarily do not have to depend upon your neighbor wanting to give you the stem cells. In fact, you can be your own donor at one time, right? So this is a doable thing. It is no longer a fiction. So you can be your own donor. So what is the biggest thing that you're going to do? You're going to avoid all the problems of getting a cell from somebody else. So when I go to the medical aspects, there are a lot of problems of getting or borrowing cells from somebody, right? From matching to rejection to hundreds of other problems, right? So I'm now coming to the next level. I also told you that amongst the adult stem cells, 10 different sources or 20 different sources from where one can study adult stem cells. The neonatal stem cells, or the cells coming from the embryo, the perinatal tissues like umbilical cord tissue or cord blood or placenta, and all those are all very good sources. A researcher can choose to work, and many have worked on it. I also want to tell you that mesenchymal stem cells is one other tissue from the perinatal tissues or even from an adult tissue, which is well worked out. And it has been proven that these cells in the laboratory, in the hands of an expert technologist who knows how to be able to give the right signal. When I mean the right signal, I mean from the embryologists, we know that if I give a certain cue, certain clue, certain hint, certain milieu to a certain cell, I know that I can gradually convert it and move it to a neuronal line, or I will move it to the gastrointestinal line, or I will move it into the cardiac line. So I am under the impression that the students who are listening to me have got an idea that it is possible for the researcher to actually manipulate and maneuver your path forward to be able to say that I know I'm working on these cells. So assume for a while that these cells have been manufactured by you. Say you have produced about 20 million cells in the laboratory, in the tissues. What do you do next? Are the questions that you're going to answer. Before that, the mood question. So if you have cells, how easy it is or how tough it is to be able to take it to the patient. And how is it work? How does it work? So there are more than one mechanisms by which we believe that stem cells work. So the whole lot of experiments, whole lot of research area, all of the researchers world over are now trying to prove that after having produced these cells have to show that they have done what they are supposed to do. And have they done what they're supposed to do? And are they continuing to what they're doing, what they're supposed to do? Or will they do it only for about a couple of minutes or an hour, few hours, or then they die? So are they going to be doing it continuously? So these are the questions that come in all of us, right? So let's look at what are all the probable mechanisms through which these cells could work so that the concept of regenerative medicine, of being able to deliver treatment to the diseased or to the disordered or to the lost cells, right? So we believe that the cells, now you have produced a certain group of cells, and these cells, we believe that they can actually help homing in. These cells, probable mechanisms are, they would actually initiate, they would initiate a series of cascades. There are so many chemical cascades. Like we have cycles, we have so many cycles, biochemical cycles. We have series of cascades. These cascades are extremely important, which normally go because there is a genetic predisposition. There are certain genes that govern the movement of these cascades, right? So these cells, by virtue of the fact that they are stem cells, by virtue of the fact that they have certain characteristics, they will start initiating certain cascades, and they would actually initiate endogenous repair, right? And probably they will also revascularize. Suppose there is a situation where there are no blood vessels or blood vessels are blocked, you will form new blood vessels. It will also reduce immune attack. So suppose there are certain cells which are actually destroying because of an immune attack. There are certain cells getting destroyed because there is an autoimmune destruction. These cells probably will produce some kind of a curtain, some kind of a cascade, which will prevent or reduce immune attack. It will also tip the balance towards repair rather than scarring, rather than fibrosis, etc. So these are all the things which we would like to avoid, and therefore the stem cells work probably through the mechanisms. Now, therefore, now the adult stem cells are believed to be the paramedics of our body, because I told you that adult stem cells continue to work throughout our life, right? So they secrete a lot of factors. So these cells are probably going to initiate secretion of factors, which will enhance or motivate or push or actually escalate the level of vascularization, etc. It is also believed that they awaken the dormant stem cells. Probably they also allow new tissue formation. So therefore, adult stem cells which are going to be given to a certain person, to a deceased organ through some methodology, will probably increase the blood flow to an organ where there is a damage to the blood vessel, or it will also promote plasticity. One other important thing, it will also convert the cell into the cell type that you are much wanting at that point. Also, it will also bring about de-differentiation of the stem cells. Here is a small word of understanding I just wanted to say. These stem cells almost try and go back to earlier stages, like almost it goes to a something like embryonic stage, and it will try to give a cue to say that, hey, come on, let's form some new type of stem cell here. And so these kind of de-differentiation is also one of the proposed mechanisms of action of these stem cells. I already told you that stem cells can transdifferentiate. So if you give a mesodermal cell, it can still form an ectodermal cell. So this kind of primitive conversion is possible, has been proven in the laboratory. It has to be proven in the animals. It has to be proven in the adults later during clinical trial. The next important possible mechanism is cell fusion, a very important mechanism which is believed to work. For example, when you donate healthy genes, these healthy genes probably will replace the faulty genes in the infected area. It could correct the defective genes, etc., which is one of the mechanisms by which they believe that in certain skeletal muscular deficiencies, they say if at all you give cells which are in normal genes, it will form a fusion. And these fused cells will actually form single cell later on. Sorry, is there any disturbance?
You can carry on.
Yeah. I hope I have some time, right?
Yes.
Yeah, because I still have a little ground to clear. Okay, so so these are the probable mechanisms. So I said cell fusion is a very important probable mechanism by which healthy genes will actually fuse, or it will replace the defective genes. And it is for us to prove how can you track these cells. Suppose you give stem cells to somebody. How do you track them? How do you see that it is converted? How do you see that it is maintained? How do you see that it is functionally integrated with the defective area of repair that you want to repair? All these things are what forms the bread and butter of all the researchers. The bread and butter of these researchers, they are wanting to be rewarded with all their findings. These are the different research that is going on. How to do it in animals, how to translate them into human beings. Because we can't do certain things in human beings. So we have to convert or do all these in small animals, show that it can be translated upscale to a large animal like us, large human being like us. So that is what is where we have to actually prove ourselves. The other most believable mechanisms by which these cells work is through their paracrine function. As I said, because stem cells secrete certain factors, there are certain factors which govern the conversion of cells in the laboratory, in the human body. It is possible that this is one of the most believed mechanisms of action. So it will tip the balance toward repair instead of doing a scarring or forming repaired, non-functional tissues. Now, therefore, it has been proven through experiments that angiogenesis or formation of blood vessels through what is called as VEGF, vascular endothelial growth factors, FGF2, human growth factor, and VEGF-FR2, etc. So these factors have been proven, they have been documented, and these factors, therefore, are indirect surrogate markers through which you can actually now, it is possible that somebody else will try to form VEGF and try to give VEGF instead of giving stem cells, because stem cells has its own set of problems, and therefore it is possible that once you prove that stem cell has secreted VEGF and VEGF has given rise to formation of blood vessels, it is possible that you can actually work on VEGF, right? Also IL-10 actually decreases inflammation. So if you have been able to link stem cell to IL-10 to decrease inflammation and therefore to repair of a certain situation or correction of the wrong, it is possible that some researcher can work on IL-10 and say that IL-10 production can actually prove very, very useful in clinical situation. The anti-apoptotic function is extremely important. It is working through improved chemotaxis. So all these are reparative mechanisms that are in play even today. All we are trying to show is link stem cell role and trying to show that we may be able to give a few stem cells. Now, when I say a few means it's like trillions. It is not 1 or 2 or a million or a billion. It is in trillions of cells that you will have to give for a certain reparative mechanism. Now, therefore, these cells are believed that increased DNA repair, increased antioxidant function, increased HGF function, and all the homing in brought about through SDF, SCF, IL-8 are all the cumulative ways by which our stem cells are going to work and make regenerative medicine truly a reality. So this remodeling is what is very important. Now, when you have cells in place, when you have cytokines in place, it doesn't happen that you can just give a trillion cells to somebody and then it will just start forming. You have to give them in the mechanism, in the way in which it is to be presented. So it will require some kind of a extracellular matrix. So that is where tissue engineering comes into play. So you will have to have certain molds, certain 3D mechanisms by which you will be, you just can't give it as an intravenous and you expect a heart to form, right? So whatever you conceive has to be later converted into a mechanism by which it can be acceptable for clinical consumption. So therefore, for those who are now wondering, where are we? We started with cells. We started talking about what are the probable mechanisms. And therefore, what is it as students? So that means somebody should understand that this branch of medicine actually engages or works with living cells, right? These cells live. They have to be stored in liquid nitrogen. They can be stored for a very long time. It can be your own cells, it can be from somebody else, right? And therefore, I can also work on an extracellular matrix like a collagen or collagen-like, or it can be something like, a tissue engineered construct. So suppose I want to give it in the form of a skin, I can't just pour the cells on the wound and say that it will form skin. I have to give it a certain scaffold, right? So the scaffold mechanisms. So all these scientists, all the biologists who are going to work here, these are the people who are going to work on cells and the around the cells. So this is an extra cellular matrix development. So the tissue engineers are also going to be part of regenerative medicine. Most important, communication. So there is to be a handshake between the cells, the scaffold, and the inner self. So with the main parent organism, the parent cells. So this communicators, this handshake is brought about through the signaling systems. So the signaling system consists of so many growth factors, not one or two or five. It's like hundreds of them. And there are those cytokines. These are the cytokines through which they work even today. And you and me, in the future regenerative medicine experts who are sitting in the audience, are going to decide their future. If at all they feel they are interested in this, they could choose to work in and around these areas. So that can form their future, right? So I'm now going to define regenerative medicine as a new way of treating diseases or injuries, excuse me. A new way of treating the injuries and diseases that use specially grown tissues. As I said, we know that they're all made in laboratory, and we can make even artificial 3D organs. It includes development and application of innovative medical therapies. And also this branch of medicine will include all those mechanisms that will reconstruct tissues. It will make tissue engineered constructs or catalyze self-renewal. That is, it will promote self-renewal of parts of the organism or the entire organism, whatever you're working with. So regenerative medicine, therefore, is a huge area. So you already have on your plate so much to know. And how much will you learn in one talk? You're not going to be able to imbibe everything. But I just wanted to give you a promise to give you a flavor. Now, what are the unmet medical needs? I'm just moving, therefore, now to the clinical side. I can't help talking about being a physician myself. I know that we have done a lot of clinical trials on some of these areas. Unfortunately, they have still to go as a prescription product because it has so many steps to follow, so many steps on the ladder. So in the area of neurology, cardiology, and endocrinology, you have any number of patients waiting for a permanent cure. Today, some of these disorders are just under control. Diabetes is just controlled, right? It is not cured. There are people who are doing insulin secreting islet cell transplants today. There are people who are doing lung transplants, liver transplants, but a brain transplant is not yet heard of. But there are selective organs, like for example, in the case of Parkinson's disease, you may be able to use a group of cells that actually produce dopamine, which is the defect. So you can correct that defect. It is doable. Suppose I give mesenchymal stem cell to a person who has got a spinal cord injury. What is the meaning of a spinal cord injury? I understand that I want to convey to you that a person whose spinal cord is injured because of an accident, because it has been chopped because of an accident, he's on a wheelchair, his lower part of the limb is not functional. So because we believe that neurons don't regenerate on their own, but it's not the case, I am going to say that if I give you mesenchymal stem cells, which will actually produce neuronal cells there, which will produce some neurons, which will correct the defect, it is possible that I may be able to do. So clinical trials are going on using various cell types. I told you about Dr. Thompson's experiment in 1997. He did the first embryonic stem cell. After that, a company called Geron invested close to $60 million in doing research on spinal cord injury using embryonic stem cells. Unfortunately, that stem cell clinical trial did not see the light of the day because there were some side effects. Now I'm coming to tell you what are the side effects. Embryonic stem cells, because they are extremely versatile, they can produce all cell types, right? You cannot produce selective cell types. So where you want to produce neuronal cells, suddenly you can produce a heart, or you can produce liver. Sometimes you can produce tissues that are unwanted. So that's why the undesirable effect, what is called as teratoma formation or a tumor formation, made up from cells of the undesirable origin, comes up. So spinal cord injury today are being studied only by using cells of the adult cell types, right? Not the embryonic stem cell types. So on and so forth. So in cardiology, there are so many clinical trials going on. In diabetes, n number of research papers are getting published. And so I just want to tell you that clinical research is ongoing. Now, as far as degenerative disorders are concerned, you will see large number of papers. It is believed that diabetes, Alzheimer's disease, cardiovascular disease, a lot of inflammatory disorders, they are all degenerative in nature. So it is possible that we can regenerate them. So you will see a chunk, 70 to 80% of clinical research papers, clinical trials that are going on are all in the area of cardiovascular research, diabetes, Alzheimer's, and large number of them even for cancer. Cancer research is going on, but stem cell research in cancer relatively lesser as compared to those that are being focused by way of degenerative research. Coming to a very important stem cell type called induced pluripotent cells. It's a very, very versatile cell. What is it? So the word induced means I am going to induce. What is the meaning of pluripotent? I'm going to make it really pluripotent. I'm not going to make it totipotent. I'm going to make it pluripotent. So I am going to make it as potent as those cells that came from the inner cell mass of the blastocyst, right? And I can have unlimited supply of this. So where are these coming from? These are coming from adult stem cells. So what did the researchers do here? The greatest research that happened, of course, it must have cumulatively, it must have happened over several years. In 2006, Yamanaka and his group in Kyoto, in Japan, published a very seminal paper on what are called as Yamanaka factors. What did he do there? The simple experiment is that he produced stem cells by taking adult stem cells, any somatic cell, say in this case skin cells, and he told the skin cells, go back to your one origin. So go to your embryonic stage. So go back in your stage. So it is called reprogramming, and go to your different undifferentiated stage. So go back to as if become like an embryonic cell, right? So once it became like an embryonic cell, by giving it backward cues. So there are those cytokine factors which will go backwards, which will make it go backwards. Once it goes backwards, it is as if you're having embryonic stem cells in your hand. And then you give the cues, you give the signal for it to produce either a stem cell, either a liver or a pancreatic, an insulin secreting beta cells, or you want to give whatever cell you want to manufacture. So it is just like taking a cell from your own body, making it go backwards. After you go backwards, you produce the cell of your interest. And by using these Yamanaka factors, these therefore will remove all the fears of ethical questions, the moral questions, the availability questions. Most importantly, I want to repeat, it is coming from your own body. So there is no question of rejection. It is not the totipotent coming from that embryo, and therefore you are not going to be able to form all the three cells. You don't form teratoma, you don't form tumors. right? Therefore, you don't require immunosuppressions. You may be able to use it in the clinic more usefully, more gainfully as compared to most other cells, right? So these offer pathways to offer adequate autologous, I am using this word, autologous cell replacement therapies. So your own body can donate your own cell. So the question of whether who can be your donor, you can be your own donor. If you have an iPS cell, if it's still in experimental stages, we still do not have an iPS which has reached the bedside. It has not come to be, mainstay treatment. But I just want to tell you that it is going to be the next milestone. It's going to be the next most the greatest advances in science today. So the challenges are there. Nothing comes without any challenges. But here, so far, there are scientists who have used viruses which are carcinogenic, which can form tumors, and these are getting replaced by non-carcinogenic tumors, or by non-carcinogenic viruses, or by usage of some other chemical forces, or by using some physical forces, or by using certain other easier methods, right? So these are this kind of reprogramming, as it is called, is now being studied by large number of researchers, and there is also research even in our own country. People are working on iPS, but we have yet to know whether it is anywhere near even clinical research. So induced pluripotent cells, I told you about the iPS technology, that is Yamanaka's laboratory in Kyoto, Japan. And these four specific genes are monitored or are being manipulated. And then they convert these somatic cells into pluripotent stem cells. And this became, therefore, the most important recipient, or he deserved the 212 Nobel Prize. And therefore, John Gurdon, who worked with him earlier, and Professor Yamanaka, they got the Nobel Prize for this in 2012. So this is just to tell you a schematic representation of iPSCs, and how it can have a conventional approach, or you can have a non-conventional nanotechnology approach. So these approaches are all the modern day research. You can decide on which line somebody can work on, and all these type of cells are possible. So you can actually form like an organoid, you can form tissues, you can use it for drug screening, you can as if use it like an embryo, you can see whether a liver which is produced in the laboratory is allergic to certain drugs, or whether it becomes toxic to her. So I'm not touching much on it because for want of time, but there are lots of applications if at all you touch this iPS as a tool for research, right? So the scope of services in this discipline, in regenerative medicine, besides what we already spoke about, so therefore somebody can actually be in the therapeutic segment. So somebody can be a doctor, somebody can be a paramedical, somebody can do clinical trials, somebody can be on these areas like tissue engineering, somebody can do working in the upscale of production. So somebody is not happy with just producing a million cells. You will have to have certain systems and processes to produce trillions. So you can be in production, you can be in quality, you can qualify some of these cells. You can be a banker, you can bank iPS cells. Now, when I say iPS cells, it can be normal iPS. I can bank cells from abnormal iPS cells. Like, for example, I can make iPS cells from a person who is already having a disease, because then these cells will carry the disease gene, and therefore it becomes a very important source of your research tool, right? So you are having a cell in your laboratory with a disease, and you know how to treat the disease, or at least you know how to work, right? So it forms a starting material. So these are all things that you never had in the past. So you have iPS with a certain genetic abnormality. You have a certain iPS with a certain proposition to cancer. You have a certain iPS cell from a person who already has a cancer. So you can study all the behavioral pattern of these iPS cell in your laboratory. So we talk about toxicity. So you can produce any number of tissues in the laboratory and then give it as and pretend that it is a miniaturized organ. Otherwise, what would have happened is that all the drugs that are produced in the country, in the world, has to go through certain toxicity trials, right? So you have to subject an adult or an embryo or an animal to all these studies. So it forms a new paradigm shift. Your thought process will change. You will think in a different direction. So there are people, so there are so many areas for anybody to work on. So for want of time, I think I will not touch on those because I want to take some questions. What about our country? In our country, we have at least a dozen, we had a dozen banks, about 12 to 14 cord blood banks, and then they started banking mesenchymal stem cells. There are other government agencies which are in research, about 12, 14 of them. And the Indian ICMR guidelines, the first one came up in 2013. And those guidelines will tell a researcher how to work with stem cells. And then regulatory framework is being laid down. It's still just being laid down. So we have a guideline for how to run a clinical trial, but it is always work in progress. So what is yesterday is not today. What is today is not tomorrow. So we always find faults with what we have now so that we can improvise. And there are a large number of committees who want to be very careful with stem cell, because as much as stem cell is very interesting, it's a double-edged weapon, because there are two sides of the coin. You get the bad, you get the good. You get both of it together. So you need to be very careful that somebody is not misusing something, right? If I say that I'm producing liver, I'm also giving some side effects. So you need to be extremely careful and wary of the fact that it could be misused. So you have to be very vigilant, very watchful of all that. What is the status of research? Research, I told you, isn't going on in many areas, but what is research? What has landed in the market so far is only some skin products. We do not still have a treatment. Of course, I told you that cord blood transplantation is the only one that is already an approved way of treating hematopoietic or blood disorders using cord blood stem cells. But apart from that, there is no approved treatment yet of a stem cell. But stem cell based some kind of products which have come, adult stem cell based products have come in the market, but they are, I don't know whether to call them adult stem cells, but these are some of the, say, alloderm and epigraft. So there are some epidermal cells, which are being used as a transient treatment when somebody is going through a skin transplant for burns, etc., etc. So besides skin, nothing much in the market. Hematopoietic stem cell is going on as a regular practice. It is hematopoietic stem cell transplant using cord blood is not as much as the way bone marrow is being used, but it's a very well recognized way of treating hematopoietic disorders. So we have challenges, lots of challenges, not one. How to scale up, how to make cells, how to make it available in the right platform. As I said, I can't just tell somebody take 1 trillion cells and you can treat somebody. It has to be in an acceptable manner. You have to give it. And all people who are in research are all under pressure to commercialize. Because whoever has invested money wants to see that the treatment has to come out, but it cannot be, because we are not playing. While we are trying to play God, we cannot play. In reality, we can't just give it away to somebody without it going through all the life cycle, all the clinical trials, the phase 1, the phase 2, the phase 3, and whatnot. So it has to go through all that. Before that, it has to go through all the preclinical experiments. It has to go through all the research experiments. It has to go through stability. It has to be peer reviewed. Then alone, this product will ever go to the bench or a bedside, right? So it is still a subject of great politics, and because there are a lot of questions that are yet to be answered, whether it is going to be funded, how many people are going to fund it, what type of research funding, how much of clinical research funding is available, is a question we can answer later on if there are questions. What you need to be very careful, as much as it is very exciting, very interesting, there are a lot of scientific flaws. What somebody has written in literature may not be repeatable, or I can't reproduce it in my own laboratory sometimes. So you need to be very careful before you say that you have been able to do something. And don't make an overstatement of its potential, because when you say something, somebody comes up with some hope. So we don't want to sell hope beyond a certain point. You know that there is a potential, but be very careful of even the underlying harms that could be, because we have seen it over the years. And do not ignore any ethical issues that may come up. So don't tell somebody that I am going to promise that your liver cell failure is going to be treated. You give me so much money and I'm going to do this and I'm going to have. So these kind of statements, one needs to be extremely careful. So even if you come across somebody who is making those statements, I think you need to be very careful, very wary. And that is what our regulators are doing. They are extremely mindful of what is being advertised, at least in this country. What are the claims that people make? So clinical trial investments are really needed here in terms of money, in terms of resources, in terms of people wanting to wait for a long time for correct results to come out. If correct results do not come out, we do not want flawed experiments, we do not want wrong experimentation, etc. The regulatory framework is under construction at all times, and we need a roadmap. So if you are a researcher, you need to know, if I start in 2015, can I have this product in 2019 if I did 1, 2, 3, 4, 5, 6? So that kind of roadmap is yet not clear. So if I did not know, I won't just go aimlessly, right? There are a lot of quacks in the world. There are people in the internet. If you go to stem cells, people will tell you so many golden stories, and you will be minus your bank balance, but you be very careful before you get blown in by the claims that they are doing. You need to be extremely patient, because I know some of us have come across families. As a doctor, I have come across hundreds of thousands of patients with autism, with spinal cord injury, patients with Alzheimer's disease, patients with rheumatoid arthritis, patients with problems which are like genetic disorders. And they always look forward to you as if you are the ultimate, but you need to be very, very sure of what you're talking to the patients, right? So the experiments have to be extremely robust. They have to be repeatable. You need to be mindful of these before you. I'm telling you these because when you read an experiment, or when you read, or when you discuss, our young minds should not be carried away by what somebody is talking about. So resources, resources, a lot of money, a lot of patience, like-minded people have to come, and good intentions are there. But with good intentions, you need to have a lot of political, lot of governmental backing, and lot of these experiments come only with a lot of collaboration. I, sitting in my own room, cannot do anything. So you need to handshake. So there has to be a physicist, there has to be a person expert in collagens, a person expert in reagents, a person who is expert in reading machines, a person who is very good at laboratory, a person who is good at production. So this kind of handshake is going to make this research really, really meaningful. So I think I've tried to convey to you. Before I just say, that's it, just want to say, therefore, embryonic stem cells, if I just want to make an analogy, it is like alcohol, very pleasurable looking, but it has its own side effects. But if I take induced pluripotent cells, they are like Coke, gives you a kick, small side effects, but still has got a lot of potential. It gives you all that a drink should give. But if you talk of adult stem cells like mesenchymal stem cells, or adult stem cell coming from tissues, your own fat cells or whatever, those are all like your homemade fruit juices. So they're really dependable, trustworthy, very healthy, no side effects. So at least, I'm just trying to keep it light, and just want to leave you with that analogy. And I hope I have been able to convey at least 50% of what I had planned to convey. If there are still some confusions and some questions, I'm really happy, and thank you for your very kind attention.
Thank you, ma'am. Million sincere thanks to you for the sea of wisdom and knowledge you showered us with your amazing presentation. Thank you so much, ma'am. I hope every listener have enjoyed this amazing presentation and benefited with it. Now, ma'am will answer some questions. So participants, please be sure to type your questions into the chat box. Ma'am, can we go ahead?
Yeah, please.
Okay, ma'am. And the first question is, which is the best place in India for doing stem cell research? Which is the best place in India for doing stem cell research?
Oh, okay. I'll answer the question a little differently. There is nothing like a best place. There are a lot of stem cells people work on. For example, some people work on stem cells from the eye. Some people work in brain. Some people work on cord blood. So different organizations have a different focus. Okay. So say suppose somebody wants to really have some goal to work on retinal stem cells, wants to treat macular degeneration, that blindness that comes with age sometimes, or with diabetes, etc. So he can go to places which are invested in this. Like for example, as an example, LV Prasad Institute, Shankar Netralaya, they all have their research wing. Likewise, you have in Pune, in Bangalore, in Delhi, there are governmental institutes which have a reasonable amount of research investment. So people should try and get to know more about them from their individual websites. And if their goals are matching with the goals of the researcher, I think you should try and attempt that. So, and most of the cord blood bankers, which are essentially private companies, their research is limited to working with sometimes cord blood mesenchymal stem cells or cord tissue mesenchymal stem cells. So they don't go to all the organs. So the rule of the game, unwritten rule of the game, is that there are some companies will focus on only on certain cells, and some companies will focus only on certain tissues. It's like that.
Next
Have I
question
answered
is,
the question? Yeah.
Thank you, ma'am. The next question is, how can we face the ethical issues and controversies when we carry out research in this field?
Oh, very interesting. So, see, fortunately for us, the ICMR has come up with guidelines, right? So, as I mentioned to you, the 2013 was the first one. We learned a lot of things, and then the next versions came. One more version came. It tells you who can do how, who can do what, and who can do how much. Okay, so suppose you are a researcher sitting as a part of, say, a company X, and if you want to do embryonic stem cell research, then there is a way by which you need to apply to the ICMR will guide you what are the requirements you need to have, what should be the laboratory, people, raw material, blah blah blah. And once you comply with that, you will be able to do that. So is the case with adult stem cell. They will tell you what to do if at all you go and approach them. So ICMR is the first body, because that is the head of, they govern research in the country. Whereas as far as regulations are concerned, how to give it to the patient, whether it is good enough to be given to the patient, that body is called as the drug controller.
So next one is, can stem cells obtained from one person be used for treatment in other person?
Yeah, that is exactly what I mentioned. I hope I was clear. I was audible, right? I'm sure I did mention that. So until recently, we used to think that, say for example, cord blood. We are giving cord blood and cord tissue. We are keeping in the bank, thinking that it is for the baby's own requirement when the baby grows up, right? But now we have proven that adult stem cells have got a characteristics which is so unique that it does not get rejected. The adult stem cells, they do not get rejected. Okay. So when that rejection is not there, I can borrow from you, I can take from my neighbor, I can take from anything. It is not like bone marrow blood cells. It is not like the CD34. I mean, certain adult cells called mesenchymal stem cells, which I spoke about, are themselves immunosuppressed. They do not get rejected. So you can take from oneself. I want to answer one more as a corollary, or rather an extension to this answer, is also the fact that if at all you have a big body, you have so many organs, You have so much of fat. You can be your own donor. You can take cells from your own fat tissue, which you can very generously donate. And then some researcher can take out your own mesenchymal stem cells. And you can use it for yourself. So don't worry about even buying from somebody else.
So, ma'am, the next question is, can we use embryonic stem cells in fibroid treatment?
So I told you that embryonic stem cells is a no, 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 no. No embryonic stem cells for human use yet. No, because not only of the ethics, because it has a tendency to form a tumor which will have all kinds of stem cells, because it is very, very, very potent, I told you, right? So you cannot use embryonic stem cells for any treatment whatsoever. If somebody has used it in India or wherever, it is wrong. There is no approval yet for anywhere in the world for embryonic stem cells as a treatment for anything yet.
Okay, ma'am. So the similar question is that is there any chance that they can themselves become a cancer cell?
Yes, that's what I said. If there are some cancer amongst the stem cells, if there is a cell which is cancerous in nature, it is possible. It can, yeah, because its growth is not controlled, right? Suppose there is an abnormal gene in one of the cells, and if you remember, I showed you a slide where one cell will produce a daughter cell and a progenitor cell. It is possible that this cell will keep on forming more stem cells. One cell will form two stem cells, and then that has got an abnormal gene. So yes, that possibility is there.
The other question is this mesenchymal cell transplant convenient for all those who are affected with spinal injury?
Yes. So mesenchymal stem cells, it is potentially very useful. It can be, unless we finish clinical trials. Now, clinical trials, for example, means I hope our audience, young audience, understands this. Clinical trial means trying it on actual patients for a long time to see whether the results are good, and then we will do it on large number of patients, right? So those clinical trials itself take anywhere between 1 to 4 years sometimes. Okay, so after you do those trials in those many people and then follow them up for a long time and say that yes, mesenchymal stem cells are very useful for spinal cord injury, that kind of thing has not happened. There has been one trial that we have done in the past, which was a very short trial, just about 20 odd patients. It should have been continued by doing it on 100 patients and then on 300, because there is no shortage of patients in our country. It should have been done. But so till you do all the levels of clinical trial, it is not available to you and me today as a prescription drug. So nobody will prescribe, okay, give 1 million per kilogram body weight for the spinal cord injury patient, inject it here. No, nobody will give you that yet, not yet. But yes, there is a potential. It is eminently possible. It will happen soon.
Okay, and we expect the treatment will be soon employed. So, is it possible to cure lupus through stem cells?
Sorry?
Lupus, can we treat lupus through stem cells? I think.
See, the same thing. If at all we are, it's a skin disorder, right? It is an autoimmune disorder. So it is believed, yeah, it means our own skin cells, certain component of the skin cells are behaving as foreign to us. So they say that if at all we inject, but how will you inject? Where will you inject? Theoretically, it is doable, but I really don't know till you do it on actual patients, 100 or 200 or 300 of them, with the approval of the regulator, with the approval of the drug controller's office, after you show your proof in research. You have to show laboratory research. After that, you should do the human research.
Can we apply stem cell for overcoming the current situation? Can we use it for COVID-19 treatment, stem cells?
Oh, that's a very wonderful question. Thank you for raising that question. I wanted to tell you that, but in fact, one trial by one company here, which is called as Relabs, got. I was a consultant until recently to that company, and uh, the uh, the clinical trial was approved. It was to be done only on a few patients in a phased manner, in patients who are having, intermediate type of COVID, and a lot of work was going on towards getting, ethics, approval of the ethics committee of the respective hospitals. So I'm not sure whether the trial has started, but an approval had come up.
Okay, ma'am.
Yeah.
So the other question is the mesenchymal stem cells only used for spinal injury? Rather, can we use them for any other defects, any other diseases?
Oh, I thought I discussed that actually. I told you that it can be used for so many, be it for diabetes, be it for a cardiac disease. It can be used for, healing, non-healing ulcers in diabetes. You must have seen people with ulcers, diabetic ulcers, that's called. So it does very well in diabetic ulcers based on our initial experiments. We have done a few patients in the past, and this has to be continued to be able to, till you study in a certain large number of patients with in a proper manner. This is called as a clinical trial. So this trial has to be done, and unless you do a clinical trial, you cannot say that it is ready for human use.
Okay, ma'am. So how stem cells can boost immunity in a person?
Yeah, it is. That is what people write in many websites and things like that. I don't believe in. You can't just drink some stem cells, or you can't take injections every day. And by taking some one injection, no. In the case of COVID, it was planned, or it was postulated that COVID will actually, the presence of stem cells, the mesenchymal stem cells, if it is given, will actually prevent the cytokine storm, because it suppresses, it suppresses the storm. So when I see suppressed storm, that lung component, after the first phase, initially after the 7 or 8 days after the COVID infection, you get the respiratory problem, right? You can't breathe when the oxygen level goes down. Person requires some external assistance to breathe. So at that time, it is believed that the lungs are full of cytokines, and that is how the lung goes into a state of shock. Lung almost becomes like a stone. It doesn't it doesn't respond to the normal requirement. So at that time, because there are so many viruses are promoting the cytokines, it is believed that the cytokine storm can be inhibited or prevented. That was the basis of that first study, which I hope will start very soon.
How will biotechnological applications will expedite stem cell research in present and future scenario?
Can you read that question again, please?
Oh, sure, ma'am. How will biotechnological applications how can they help in stem cell research in present and future scenario?
Always. Without biotechnology help, there is no progress at all. From day one, the moment I take out the cell to show that the cell is a cell, to show that it has surface markers, to show that it has a certain gene, unless I use the flow cytometry, unless I show certain dissecting experiments, unless I show that it is expanding, unless I show large number of cells, everything is that. And you have to studying the genes, right, at all stage. Because when you're studying the stem cells, you need to study the fate of the stem cells. When I study the fate of the stem cells, I also want to see that what happens to the genetic composition, what happens to what kind of cues are required, so that a cell, instead of forming the pancreas, is going to form something else. So without biotechnology, without a PCR at all stage, without understanding what type of cells, how do you extract the genetic material, how do you see whether, what is the meaning of fusion, how do you see that there is an epigenetic change, everything is biotechnology. Biotechnology is the backbone. When I say biotechnology is a very big word, because there is recombinant science, there is laboratory science. So I'm talking about the laboratory support. I'm talking about the laboratory. And coming to the specific question, if somebody has, what is a biotechnology? Suppose I tell you that X is a protein or it's a cytokine. That is the, that is the, that is a, what do you call it? That is a magic bullet that actually treats a particular disease. If I know the characterization of that bullet, I may be able to reproduce or manufacture it in the laboratory in certain other microbes. So that is biotechnology. So I can produce it in a recombinant fashion if I come to know what is that particular, what is a gene sequence, or what is the actual, if I know what is the sequence of that particular protein. So to be able to know, once I know what is the answer, I go backwards and I produce it again in the laboratory. So that is biotechnology. So whether in algae or in, candid, wherever you want to produce it.
Okay, ma'am. The other question is it possible to reverse any type of cell by Yamanaka method?
It is, yeah, it is technically any adult cells. Now people have studied maximally, they have studied skin cells, adult mature skin cells have been taken and studied. But it is believed that any adult mature cell, once you engage these technologies, it is expected to get de-differentiated. That's what is.
Okay, ma'am, it looked like we have completed almost every question in the chat box. We have covered almost everyone. So thank you, ma'am. Thank you, ma'am, for the
Thank you
answering.
so much. Thank you. I hope, because this is a virtual one, I don't even see the faces of the people to whom I talk. Then you get a feedback. Here I don't get a feedback. So it's a new experience all the time, when you talk to a computer. So I hope people have benefited, or they have understood at least some percentage of what I wanted to convey today.
Really a wonderful one, ma'am. Exactly, everything is perfect, and it's a fascinating presentation, ma'am.
Thank you. Thank you.
So,
good evening. I feel proud to deliver the vote of thanks to one and all present here. Let me express my sincere gratitude to the chief guest and the speaker, Dr. Chandra Viswanathan, for delivering such a wonderful session. Through this session, we are enlightened with the detailed knowledge of stem cell biology. Thank you, ma'am. Thank you very much for motivating the student community to study intensely the stem cell biology and to do research in this field. Thank you, ma'am. Thank you very much.
Thank you. Pleasure was indeed mine too. Although I'm not seeing all of my students who are sitting in front of me, but in this new changed circumstances, it's a new experience. And my sincere thanks to Aluva, all the organizing committee members, Dr. Susan again, and the head of the institute for being kind enough to, have me over. And I too enjoyed it, and I hope it will be a continuation of this as we move forward. We will upgrade and learn more and more of this. Thank you, and I wish you every luck in the coming days.
thank you, ma'am.
Chandra,
Madam, we will continue our communication with you. We'll continue our communication with you. Thank you, ma'am.
Thank you.
Next, I'd like to thank our principal, Dr. David Sach Matthew, for the continuous effort he has given to us in organizing this program. Next, I thank Dr. Susan Epen, whose initiative and inspirational move that led to the beginning of this webinar. Next, I'd like to thank our HOD, Mr. Shyam Mohan, whose constant encouragement give us in continuously continuing the seminar with a success. Next, I thank Dr. Serene Sarah John, whose coordination led to the successful ending of this webinar series. I'd like to thank the student community for the continuous effort they have taken in leading this program to a successful one. Finally, I'd like to thank all the participants around the globe who are constantly watching this webinar series. And I think they have benefited from viewing this webinar. And I thank on behalf of our college, all the participants who really participated in this webinar. Have a wonderful evening. Thank you.
Thank you.
Okay. Vedana?
Yes, ma'am.
Okay, continue. Carry on and let's wind up the session.
Thank you, Chandra. Thank you, Chandra.
Thank you, thank you, thank you. I hope I could meet your expectations.
Yes, exactly, ma'am.
Yeah, it was fantastic. Yeah.
Expectation.
Thank you. We'll keep in touch, yeah?
Yes, yes.
Okay, bye,
Yeah,
bye.
sure.
Bye, Dr. Susan. Bye, bye. And the party,
bye,
and
Chandra.
don't
Yeah.
forget the party.
Yeah, sure.
Okay,
We'll meet in
okay.
Chembur.
Okay, bye, 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 bye.